Hello students, welcome back to another new video on digestion of protein. So we have done the digestion of carbohydrate, and after that we have also practiced the absorption of carbohydrate. But today we will be learning about the digestion of protein. From protein food, we all know that we get protein either from the animal variety, maximum the animal variety, and also the plant variety. So animal variety of protein are more complex and plant variety a little bit simpler. Both of them helps to fight infection and they help in cell division. So dietary proteins, they are acted upon in the mouth, then they move into the stomach where the HCl and pepsin acts, and they are denatured, and ultimately to the small intestine they are converted into amino acid. We all know that all the amino acids are joined by peptide bonds to make complex protein. So amino acid is the base or the key of the, or the, you can say, the structural unit of the protein. So our main target is, before absorption takes place, we need to change this proteins, whether in the primary section or the secondary section, or in the tertiary structure, whether whatever structure they are having, into simple amino acid for future absorption. Welcome to Educators, myself Manishta Chatterjee. Keep on liking my videos as you are, and don't forget to share my videos. Try to subscribe and put the bell icon for more updates on notifications. Thank you. Coming to the protein digestion, the first step in digestion is the mouth. As we know, in any kind of digestion, it's the mouth. Now, over here we have taken an example of an egg, and how the egg is being denatured, and how the egg is being digested in your body. We will give, just we are reading through an example, you can see. So the first digestion in the mouth takes place in the oral cavity, with the help of the salivary gland and also the pharynx and the tongue. Now, what happens? As you all know, mechanical breakdown of the large pieces. Suppose egg or any kind of protein, in that instance, when we are chewing, it is broken down into smaller pieces, and the salivary gland, they are uh, helping in swallowing or passage of the food particles down to the esophagus. Now over here we are showing that how complex a tertiary and the quaternary structure is, then the primary structure and the secondary structure. So these are all the protein sources that we get. So we will learn to main know how to how we are breaking these structures, these complex structure, into simpler form, so that we can actually uh, absorb them in our body for its function. Main function of the protein digestion starts in the stomach. There are main two function in the protein digestion in stomach. First is the denaturation, and also the enzymatic digestion, that is pepsin. So we can say that in the stomach, both mechanical and chemical digestion takes place, and mechanical digestion also takes place in while the egg is passing, or while any protein is passing through the esophagus by the um, contraction and the relaxation of the esophagus. So I have shown you how complex the quaternary and the tertiary structures are. So we are main function, or our main function, is to make them simpler and to break those the bonds, the peptide bonds, the complex peptide bonds, and made them into a simple chain. So over here in the gastric juices, we have the two gastric juices. One is the acid, the normal HCl, and the pepsin, which is the gastric juice over here. They initiate the chemical digestion of the protein, whereas along with that there is a mechanical digestion where there is peristalsis. So both peristalsis, this churning in the stomach, within the stomach, along with the the gastric juices, the action of the HCl, we are coming next to the action of the HCl. They are mixing up this whole small, broken fragments of the food, or broken fragments of the protein, into a mixture more uniform in nature called the chyme. So we have also read about chyme in the carbohydrate digestion part. So here also the chyme is made within the stomach. Now, stomach has very important functions. First of all, the stomach has hydrochloric acid in it, and you know that it has a very low pH of 1.5 to 3.5. Now, what happens to the food, or especially to the protein, mentioning the protein in this acidic condition within the stomach? What happens? The protein are denatured, and the more three-dimensional structure, or the more complex structure, are converted into the simpler form or more denatured protein. So over here, if you can see the uh, picture over here, the picture over here is showing the more quaternary and a very complex structure, which is on the action of hydrochloric acid, is converted to a denatured protein, which is more simpler in nature. But still now we have not got the amino acid. It is just breaking the bonds, breaking the three-dimensional structure into simpler structure. Okay. Now, once this protein is denatured, then by the help of the hydrochloric acid, then the enzymatic action of pepsin starts. And now this pepsin, they break these bonds over here with the action of this pepsin. They are converted into very short polypeptides in this picture, as we can see. Please note the dashes over here. Just I'll try to highlight. Yes, this dash. Okay, that means there is so much of breakage. See, what was the condition over here? There was no such, breakage, just a simpler form. So the action of pepsin after the action of hydrochloric acid or denaturation is done, it is broken down further. Now the next portion, once the denaturation is done, and it is now into smaller peptides, not amino acid but smaller peptides, the small intestine takes the major role. Over here, there are many enzymatic digestion that takes place, and also in the small intestine, absorption of these protein also takes place, which we'll cover in another short video. So, in this protein digestion, what takes place? There are certain enzymes. Now, please remember that these enzymes are inactive in nature. They are zymogens, okay, and they are basically activated by themselves. Auto activation can take place, or with the help of this enterokinase enzyme. Enterokinase. Now, what happens? Trypsinogen, with the help of this enterokinase, is converted to trypsin. And once this trypsin is activated from trypsinogen, it by chain reaction acts on chymotrypsinogen to chymotrypsin, proelastase to elastase, kallikreinogen to kallikrein, procarboxypeptidase A to carboxypeptidase, and so on. So basically, they are an auto reaction, and auto reaction takes place, and auto activation takes place by this trypsin. Okay, so when the food, the chyme rather, the churned mixture leaves the stomach and come into the small intestine, maximum of the protein digestion takes place. Till now we were actually breaking the protein into smaller parts, but now it is the digestion of the protein that takes place. And all these enzymes, after getting activated, they try to break down the proteins into tripeptides, dipeptides, and also individual amino acid, which uh, help in further absorption again. Okay, so and also keep in mind that the cells of the small intestine, they also release certain additional enzymes which help in the enzymatic digestion of the polypeptides. So I hope the protein digestion is clear to you. um, go through the video in detail, and please message me if you need any other topic, because I'm going, as I have mentioned in my previous introduction, that I'm going syllabus wise. I'll also start class 11 and will try to um, do 11 and 12 at par. So I hope you are liking the videos. Please comment if you need any topic that we need a discussion on. So that's all for today. Thank you for joining educators.